Okay, so this video is going to be about the structure of proteins. And so what we're going to need to firstly go over is the central dogma of biology. And what this basically is that DNA is first replicated and then it's transcribed to RNA, and that RNA is translated to protein. And what we need to understand first before we start getting into the actual structure of the proteins is that right as it comes out of the ribosome, the proteins, they just start immediately folding. So the primary structure of proteins, it's literally just amino acids. So they're just a bunch of amino acids, and they are stabilized by these peptide bonds, which are, and it's created by the ribosome. That's all we need to know about the primary proteins, is that peptide bond stabilizes it and they are created by the ribosome. Go next. So the secondary structure are the alpha helixes and the beta sheets. So the alpha helixes are stabilized by the hydrogen bonds between the carbonyl oxygen and the amide hydrogen on the same strand. So if you can see this could be a carbonyl oxygen, this could be an amide and a hydrogen, and due to like the electronegativity, they're being hydrogen bonded and it's on the same strand. Next. While the beta sheet, it's also stabilized by the hydrogen bonds between the carbonyl oxygen and the amide hydrogen, but as you can see, they're on different strands, and it can either be parallel or anti-parallel. Parallel is the strands are all going in the same direction, and you can see they're hydrogen bonded with a different kind of strand. It could be looping around or it can come around. And anti-parallel is that these strands, as you can see, carbon on this end, not carbon on this end, it's a nitrogen, so this one's been flipped around. So you can go next. Then we can also have disordered proteins. Disordered proteins, still secondary structure like alpha helixes and beta sheets, but disordered are neither alpha helixes or beta sheets. They're disordered. They're usually less stable, and they can connect. This picture kind of gets it. They can connect the alpha helices to the beta sheets, and they can also form the termini of a protein. So this is a tertiary protein. The tertiary protein is the 3D folding of just one single peptide chain. And so what stabilizes it primarily are the hydrophobic interactions. So when it gets folded, and the entropy of water wants to hydrogen bond with other water in as many different ways as possible. So that's going to force these let's say these green parts are hydrophilic. It's going to force them into the middle so that these hydrophilic outside can be touching the water. They can also be stabilized by stuff like disulfide bonds. They can be stabilized by van der Waals attraction and salt bridges. Next. And this is a quaternary structure, which is basically just two or more polypeptide chains. So you can think of it as this orange one could be a tertiary structure, this red one's a tertiary structure, and this yellow one's a tertiary structure, and they've just mashed together into one quaternary structure. These are also held together by hydrophobic interactions. So the way this naming system works is that you can have homo and then however, or hetero. So you can have homodimer or homotrimer, homomonomer. What that basically means is homo is the same, and the second root word is how many of these subunits you have. So a homodimer would be two of the same tertiary structures combined into one quaternary structure. And so a homotrimer would be three tertiary structures stuck into one, all the same. And then you can also have a heterodimer or trimer. Hetero meaning they're different, so you can have three different tertiary structures, and that would be a heterotrimer or heterodimer.